Hi, and welcome to the next instalment of the Oxford Centre for Personalised Medicine Flash interview series, where we talk to individuals playing important roles in personalised medicine in one way or another. Today's guest is Professor Clare Turnbull. Clare is a professor of translational cancer genetics at the Institute of Cancer Research and an NHS consultant in clinical cancer genetics at the Royal Marsden NHS Foundation Trust. Clare also works as a consultant in public health medicine for Public Health England. So Clare, it's really great to talk to you today. Could you kick off by telling us a bit about your background, so your own personal journey and how you got to where you are today?
Yes, so thank you for inviting me. So I trained in medicine. Well, in fact, actually, I initially read maths at university for a few weeks before realizing that was way too hardcore. Switched over to natural sciences and then switched to medicine. So yes, so I trained in medicine, and having done some jobs in various different aspects and specialties across different branches of medicine, paused and did a master's in epidemiology and statistics at the London School of Hygiene, because I knew I was interested in how we ask and answer questions, but overall I felt my instinct was more I wanted to do that with data rather than in the lab with a pipette. So I think that armed me quite well, not least with the ability to code and analyze my own data, which then serviced me well later on doing a PhD at the Institute of Cancer Research in cancer susceptibility genetics, and just being able to analyze my own data through those epidemiological and coding skills was hugely advantageous. So yes, so I specialized then in clinical genetics and managed to weave in my epidemiology and research background with my training in clinical genetics, particularly cancer genetics, and now work largely in the area of research, but asking and answering questions around how can we use data and numbers to better inform how we manage people at genetic risk of cancer.
Lovely, thank you. So with your cancer genetics hat on, what does the term personalised medicine mean to you?
So I think personalised medicine came from distinguishing from the results of very large epidemiological so-called one-size-fits-all studies, that actually you wanted to break groups of patients down into smaller groups defined by more specific characteristics. And I think that does make sense. I think at the other end of the extreme, there's this notion that we will sequence someone's tumor and get an absolutely individualized readout of the genomic makeup of their tumor and use that to pick out a drug that works for them. And there's some happy ground in between, because very large scale epidemiology doesn't take into account the individual characteristics of the patient. But actually, if we only study one patient, we don't have enough data or statistical power to be sure that the treatment that we are proposing works for them. So I think the term personalized medicine, many people would be a little bit cautious about using that, and maybe a term like precision medicine might be a sort of safer middle ground. So trying to use the characteristics of that patient, possibly knowledge about their germline genomics, possibly in the realm of cancer, specific knowledge about the molecular makeup of their tumor, and integrating all of that information to select the treatment that is most likely to be effective in them.
Brilliant. So my own background, and that of many of our interviewees so far, has been more in the field of rare disease genetics and genomics. So how do you think that precision medicine differs between cancer and rare disease? And what are the biggest challenges surrounding precision medicine in cancer?
So precision medicine in cancer, there would be two sort of main areas. One would be cancer susceptibility, and that's largely where I work, which is using information about genetic risk and non-genetic risk, epidemiological factors, and integrating those to best predict who is likely to get cancer and who is at significantly elevated risk of specific cancers, and therefore to use that information to implement interventions such as early or more intensive screening, chemo prevention, or surgical prevention. And in that regard, susceptibility genetics is somewhat like other types of rare disease genetics. And actually, for example, within the NHS test directory, cancer susceptibility sits with rare disease. It's a lot of the same principles. There is then, of course, precision medicine as practiced in relation to how we treat a cancer. So that is looking at the molecular characteristics of that cancer. So firstly, the genomic changes, but also you might also look at expression of different proteins, methylation and so forth, and using those molecular data to inform which drugs you use to treat that person's tumour, and also to make prognostic decisions as well. Historically, there's been somewhat of a separation between cancer susceptibility genetics, i.e. predicting who is at elevated risk of developing a cancer, and molecular oncology, i.e. managing that cancer based on its molecular makeup. But they are coming together a lot more. And I think a lot of that's driven by the role of mutations in BRCA1 and 2 informing eligibility and potential response of a patient for PARP inhibitors. So we are seeing the two coming together a little bit more, with what is a germline genomic feature being an informative diagnostic marker for a treatment. So in terms of challenges and precision oncology, there was great excitement about 20 years ago with initial emergence of so-called targeted therapies, and in particular the use of imatinib in chronic myeloid leukemia, which has absolutely transformed the prognosis for that disease. And there was anticipation that these so-called targeted drugs, typically tyrosine kinase inhibitors, which target a particular signaling molecule, that these would absolutely transform the management of many different cancers. The reality has been that certainly in some areas, in particular lung cancer, these drugs have made a very significant difference. But overall, what has become evident is that Mother Nature is quite wily, and that tumours are ruthlessly Darwinian. And for the most part, if you block one signalling pathway, that the tumour will find other ways of driving its own growth, and therefore the tumours evolve and develop resistance to these treatments. So I think it is this tumour evolution which is challenging the use of the so-called targeted drugs. So actually, for a lot of tumours, we still use conventional cytotoxic chemotherapy. So yes, I think it is that the Darwinian nature of cancers, which if you try and deliver a very precise, biologically elegant, targeted treatment, the cancer finds a way around it.
So how does your research then fit in with the aims and objectives of precision medicine? Could you discuss any particularly interesting cases or programs that you've worked on or you're currently working on?
So my research largely is focused on cancer susceptibility genetics. So identifying and implementing genomic markers that are associated with elevated risk of specific types of cancer. And part of what we do is look for the new associated genomic variants, and part of what we do is try to better understand the pathogenicity of variants and the level of risk associated with genomic variants in known genes. And then the third element of what I do is actually try to develop better pathways by which we can actually deliver this testing within routine clinical care. One area in which I work is variant interpretation. And many people think it is a sort of solved problem that we can sequence genes, we have the bioinformatics by which to call the variants from the sequence. And then the third part is understanding which of those variants are pathogenic and disease associated and which are innocent and benign. And actually, we are just in our infancy of understanding this. And actually, a lot of the tools and the evidence that we've used historically, we use rather poorly, and we don't actually understand the association of that evidence with disease pathogenicity. And so a lot of our work is around trying to unpick how we can use evidence to appropriately score variants which are indeed pathogenic as pathogenic and ones that are benign as benign. Because if we get that wrong, then we manage patients incorrectly, and the whole value of the genomic analysis, the whole benefit has actually been lost if we treat people who don't have high risk variants as if they are high risk, and vice versa. And then another program I'm working on, which is also CR UK funded, is called BRCA Direct. So again, we are looking for new genes associated with breast cancer, but the reality is actually BRCA1, BRCA2, and to a lesser extent PALB2 are the big hitters. We are not going to find any other genes anywhere like those ones. And it essentially, from the number of BRCA1, BRCA2, PALB2 mutations that should be out there, we have found somewhere between 3 and 5% of the people who have them. So it seems inappropriate, it seems ironic or misplaced to spend a lot of energy going and looking for new genes, which we know will have a much, much lesser effect, when we are currently doing very, very poorly at finding the patients or the members of the population out there who have pathogenic mutations in the big hitter genes. And one of the reasons that we are not very good at ascertaining these people is we historically have done it through a clinical genetics model. And that is saying, oh, we'll only do genetic testing in people who have a very, very strong family history of cancer and who make their way and gain a referral to clinical genetics based on that family history. And I think what we need to do is go out there and say, actually, let's test BRCA1, BRCA2, PALB2 in all comers with breast cancer, and potentially go out the next step and go into the general population, maybe test anyone who has ever had breast cancer, anyone who's got a first degree relative with breast cancer. And in due course, potentially, if we have capacity, test or offer a test to all women at age 40, because knowing about there is good health economic data around, these genes are sufficiently high penetrance for breast and ovarian cancer, that knowing about them would change your management in regard of having screening, potentially preventative surgery, potentially chemo prevention. And also when you find one person, you can then test all their relatives. So the BRCA Direct study is looking at using digital platforms, saliva testing, for getting that process of testing, pre-test information, consent, return of results, get it all very, very streamlined and high throughput. Because actually the lab testing can be very high throughput with NGS platforms. It's just having the clinical workflows to match that. So that is a program of work that's very much related to delivering precision medicine and actually delivering tests on the genes we already know, we've known about for many years, but actually making sure we go out and find the people who have them, rather than looking for new genes, which actually may be of very modest effect or extremely rare in the population.
Thanks, Claire. That's absolutely fascinating. I'm really interested to see where this programme goes, I guess, in the future. So could you tell us a bit more about your work as a consultant in public health with Public Health England?
So I'm an honorary consultant in public health, and that's through recognition of the various elements of work I've done throughout my research career. So my work with Public Health England, I work with the genomics team there, and we have a programme of work, CanGene CanVar, supported by CR UK, where we have data submission from all of the different labs across England, hopefully soon to be the 4 nations, and they've submitted all their data going back 20 years on genetic testing of cancer susceptibility genes. That we are then accruing in Public Health England, linking to the cancer registry so we can start seeing what happens to these different patients, collapsing the data down because we can then see which variants are overrepresented in the tested population, and that helps us with variant interpretation. Potentially, in due course, creating registries, and that will allow us to make sure we know who all the BRCA1, BRCA2 carriers are, the Lynch carriers. This allows us to check whether they are having the appropriate management in terms of getting their MRI scans for the BRCA carriers, getting their colonoscopies and so forth for the Lynch carriers. So actually, it's a really, really good blueprint project to see whether we can amalgamate all of our genetic testing happening out there peripherally in one place centrally, and how we would manage those data. At the moment, we're just doing this for cancer susceptibility genes, but in theory and in principle, particularly because Public Health England has now moved over to NHS Digital, which is going to merge with NHS England and Health Education England, that actually this could be a blueprint for holding the genetic data and creating registries for all different types of rare disease, with the value, of course, of knowing where all these patients are and ensuring we manage them optimally. So it's a really exciting initiative. We're the only place in the world where we are able to get all our data centrally in one place, and also then make use of these other data collections. So the cancer registry, we can link there. HES data, we can link and see what hospital activity is going on for these patients. So it really is making very good use of all of the different types of data collections happening anyway, with the end goal of ensuring we are managing our patients as best we can.
Fantastic. And finally then, where do you see the field of precision medicine in cancer heading in the next 5-10 years, say, in your field? And how do you think we're going to get there?
So in terms of cancer susceptibility genetics, my very strong feeling is we should concentrate on doing what we know is effective well. So rather than looking for new genes or testing for genes that are, low penetrance or very rare in the population, concentrate on the big hitter genes, BRCA1, BRCA2, the Lynch genes. Go out there, find all our carriers, and go and really reach out and do a lot more testing. The amount of BRCA and Lynch testing we've done in this country is very, very low. We've always had these very, very high bars for gaining your test. You have to and spent a lot of our energy evaluating people as whether they're eligible for a test or not, and scoring family histories using these sort of arcane tools to tally up whether they're eligible for a test. The lab tests are pretty cheap. We just need to develop the clinical pathways to make sure we can really open up testing and offer it to many more people. So the Lynch testing, NICE has now approved that it should be routine for all comers presenting with colorectal cancer and endometrial cancer. I think we need to move across to the equivalent of offering BRCA testing for all breast cancer cases, and potentially again roll out Lynch and BRCA testing out into, offering it to people who have had these diseases historically, and also potentially people who have an affected relative. So really go out and find all the BRCA and Lynch carriers, and then make sure we are managing them properly, because this is the archetype, this is the archetype of clinical genetics. These are high penetrance conditions for which we have some evidence-based management. So why the hell aren't we going out and actually finding all these people and managing them properly? And there's a real inclination to sort of see this, the sort of, the shiny, glittering lure of new technologies and new tests and new genes and so forth. But actually, we know that these are going to be less effective than the genes we already know about. That is a sort of a function of winner's curse. We're always going to find the highest penetrance ones first. So let's do those ones properly, rather than, the sort of, rolling out whole genome sequencing or polygenic risk scores and so forth, which are of arguably pretty equivocal benefit in the vast majority of the groups in whom they're being proposed.